to the Principles of Neuroscience with me, Sharifa. Today, we're going to be talking about neurotransmitters. Are you ever feeling down in the dumps? Ever wonder why? GABA is actually the neurotransmitter that's responsible for improving your mood. Which reminds me of a friend.
Meet Gaje, a student majoring in neuroscience. And here comes Kenu, a happy-go-lucky history major.
Why so glum, Jack?
It's my thesis experiments. They aren't making any sense.
Well, at least you get to stay inside and look through a microscope. You know, Larry was sitting outside yesterday in the rain for 4 hours, counting brackets.
He was?
Yeah. Wait, what kind of cells are you looking at again?
I'm looking at neurons. I finally got them to grow in a dish and can record from them, but my data is really weird.
You must really need a small microphone to record them chatting away.
Not sound. You record electrical activity with a small electrode stuck into the cell. Every time I stimulate them with a neurotransmitter, I get some spikes. Like this one here, it's called a trace.
So how can neurons carry electrical signals? I've heard of dendrites and axons and stuff, but it never made much sense to me. Aren't dendrites and axons are just like wires that connect to each other using chemical signals?
Can you help Gaje? What do you think the right answer is? Did you answer C? That's right. All cells regulate the material that travel across its membrane using special tools like transporters, channels, and pumps. Neurons spend a lot of energy controlling their ions. The electric signals, which are used to carry information, are based on the controlled flow of ions across the membranes. The balance of the ions is critical for neurons to function. Neurons are special in a way because of the neurotransmitters it has, which depolarize and hyperpolarize the membrane. These neurotransmitters affect the ion flow in the neuron, which may determine the direction of the impulse based on whether the neuron is excitatory or inhibitory.
Wait a minute. That doesn't make sense. I thought membrane was supposed to keep ion and stuff like that in cells.
They do, but some proteins can help molecules move through membranes. We have a lot of proteins in our cells, especially in our neurons. Some of us may even have more neurons than others.
Okay then, Brainiac. Why would ion wants to move into a neuron if you drop neurotransmitter on it?
Can you guess what the right answer is based on what we talked about before? E is correct. Molecules move across membranes through membrane potential, which is the relative net charge on both sides of the membrane. A typical cell membrane, or the resting membrane potential, is usually around 60 millivolts, where the inside of the cell is more negative than the outside. An electrochemical gradient is the combined electrical and chemical energies for a given ion. To cause changes in membrane potential and carry information, the ions must be able to move across the membrane. There are several ways to do so. One is by using a transport protein, which recognizes substrates and catalyzes its movement across the membrane. Active transport requires energy to move the solutes against the concentration gradients. Ions are considered frequent targets of these transporters. Another way to transport ions through membranes is through diffusion, which are divided into two: simple diffusion and facilitated diffusion. Diffusion is when solutes move down with the concentration gradients. Simple diffusion happens without any cellular energy. This makes it energetically favorable. The only difference between simple and facilitated diffusion is that facilitated diffusion needs enzymes or transporters to transport the solute, while still going with the concentration gradient. Different enzymes will facilitate a different amount of solutes. The KCC2 symporter is the transporter found on neurons, which facilitates the potassium electrochemical gradient to move chloride out of the cell. This protein is essential for the maturation of neurons. Channel proteins allow a different mechanism of moving ions across the membrane directly. Unlike transporters, they form a hydrophilic corridor, which allow the ions to pass without binding in a channel. These channels are usually ion selective. This selectivity is controlled by the extracellular region of the protein. Ion channels are usually closed until specifically opened for a period of time before closing again. That's why it's called gated. There are 3 types of these channels: ligand-gated, voltage-gated, and mechanosensitive.
Huh? An electrochemical gradient? Sounds like chemistry to me. So you're telling me that it works just like a diffusion of a dye in the water?
Exactly. It's not just concentrations that move the ions, but also electrical charge. Neurons are normally negatively charged on the inside. The more positive they become, the more spikes they get.
Okay then, if you say so. Then I guess you can cause neuron to spike if you drop neurotransmitter on it.
Yep. The one I'm studying is called GABA. It usually makes neurons more negative and keeps them from firing away.
So what kind of proteins that lets negative ions in when you add a chemical neurotransmitter?
Looks like Kenu is interested now. Can you help him answer the question? Remember the previous explanation about channel proteins? We know that there are 3 types of channels: ligand, voltage, and mechanosensitive. Since we're talking about neurotransmitters, a chemical signal, then yes, the answer is ligand-gated.
Okay, I get that the channels work like a gate to let ions into or out of the cells depending upon their conditions. Then what? So what happens after the ions get into the cells?
Well, a different protein channel opens and it lets the same ions move back out.
Oh, okay, I get it. When you put, what is it again? GABA into your cells, it lets the channels to open and lets the ions into the cells. So, what kind of ions does this GABA let into the cell?
Chloride. Well, when chloride enters the cell, it's supposed to make them more negative and therefore makes it harder to spike.
Fair enough. So, what's your problem?
Well, GABA is supposed to do kind of the same thing, make the cells more negative and keep them from spiking. But in some of my cultures, they're doing the exact opposite. At day 9, it seems to make them more positive and spike more. All of my other neurotransmitter samples are doing the exact same thing at the same age. Here's the data. Go see for yourself.
I see what you mean. Dina neurons are spiking when you add in the GABA. That isn't supposed to happen, isn't it?
No, not at all. I've tried this experiment 5 times already. And every time I try it, I see the same results. The younger neurons, they react to the GABA and they spike. I don't get it.
Are different neurons making different GABA channels? Different protein might respond differently.
Well, I've thought about it. I checked my cells and the exact same GABA channels are there. If the exact same channels are there, they're supposed to work the same way, aren't they?
Yeah. Can other ion pass through the channels activated by the GABA?
No, they work specifically. I even made sure to check if there are any GABA channels made here, but there aren't any. I don't know what to do next.
Well, it's a tough one, all right? Are there any different protein?
Well, only about 25,000 or so. I can't go looking for a needle in a haystack. I need to graduate by May.
Well, I'm not really a scientist. But there might be something different between them. Maybe the chloride doesn't like the smell inside the young neurons.
But ions can't smell.
But there must be something that's stimulating the neurons. But I still think that the chloride ion doesn't like it inside the cell somehow. But when in doubt, go back to the basics, I always say. Anyway, I gotta run. Will I be seeing you next week?
Not unless I figure this out by then.
Looks like Gajah is stuck in a pickle. What do you think is the first principle of the direction of chemical reaction? C is right. Free energy is usable energy, or energy that is available to do work. Every chemical reaction involves a change in free energy, called delta G. The change in free energy can be calculated for any system that undergoes a change, such as a chemical reaction. Two factors that affect the free energy of a reaction is the concentration and the charge. For uncharged molecules, the free energy is determined only by concentration. For the chemical reaction of solutes that enter the cell, delta G is equal to RT times Ln times the concentration of the solute before it enters the cell divided by the concentration of the solute after it enters the cell.
So, hey there, Genu. You know, you're actually smarter than you look.
Of course I am. How exactly am I smarter?
Well, remember how you told me to go back to the first principles? It actually worked for me. Have you ever heard of free energy?
As opposed to slave energy.
Ha! Spoken like a true historian. But actually, no. Free energy is what moves ions into a particular direction across the membrane.
Okay, so how does it do that?
Well, it all comes from this one equation.
Don't you go pointing that equation at me? I left math a long time ago and don't want to go back.
Okay, I promise I won't make you do any calculations and I'll tell you the details. So, basically, free energy is determined by two things. The concentration of the ions on both sides of the membrane.
Right, diffusions.
Exactly. So, the concentration and the electrical charge of the ion and the cell itself. Let me actually show you. If GABA is working as an inhibitory transmitter, the neuron should become more negative. This is what they are doing in the older neurons. If GABA is working on an excite neuron, then chloride must be flowing out of the cell. Follow?
Barely, but go ahead.
So, I thought about free energy and the equation. One way to make chloride enter a cell is to lower the intracellular concentration of chloride when it gets older. There are only a few proteins that can do that, so I started looking. Have you ever heard of a protein called KCC2?
You don't really expect an answer from me, do you?
Sorry, well, KCC2 is actually a symporter, and it carries potassium and chloride ions out of the cell. I don't see any other difference based on potassium, but if chloride is lower on the inside of the cell, GABA will open at the same exact channel on cell surface, and chloride will flow into the cell instead of out of the cell. KCC2 is only found in older neurons. Here's the situation. For free energy, we only worry about chloride.
So, by lowering the chloride concentration in the older neurons, the KCC2 turns into GABA and it becomes an inhibitory transmitter. Does it do anything?
Well, it seems to be important for making synapses initially. See, if the target cells don't become excited, they don't know that an axon is trying to make contact with them.
Hey, I'm all for making contact. By the way, are you free later tonight?
Sorry, Kenu. I'm an old neuron. I get excited from way more than that.
Wasn't that exciting? What did you learn from the story today? Comment your answers down below so that we know that you know that neuroscience is hard. But not so hard because of our lecturer. We love you, Miss Dina.